study more efficiently by focusing on the highest yield material. In this video, we're going to be covering amyloidosis. This is the fourth video in my series of 8 videos covering cell injury and an introduction to cancer. So when you're done with this one, I hope you will check out the rest of these videos. You can see here in the top right corner that I give the overall topic of amyloidosis a solid. Amyloidosis is not a single disease, it's a collection of diseases that result from different causes that all end up with some sort of misfolded protein. A picture that looks like this. And that's pretty much the main way to diagnose multiple myeloma, some sort of plasma cell disorder. And here, this disease creates an excess of light.
Appearance histologically is of an amorphous appearing, often pink appearing protein that is deposited in the extracellular space. Pathologists use a particular stain called the Congo red stain, which highlights the apple green birefringence of the amyloid deposited around the blood vessels and also within the extracellular space around some of the surrounding cells. Now, the term amyloid actually refers to a group of substances. There are over 15 up of the beta pleated sheet, and this is where the Congo red stain that we mentioned in our earlier slide congregates and gives the apple green birefringence.
Amyloidosis awareness. Throughout our lifetime, our DNA codes for the production of small molecules called proteins. These proteins provide the structure. When proteins are misfolded, it can impair our body's normal function. In cases, the misfolded proteins, called amyloid, meaning starch-like, take on a particular shape that makes them very difficult to break down. Because of this misfolding, amyloid proteins bind to 5 different proteins have been identified that can lead to amyloidosis. Amyloid is derived from misfolded light chain antibodies, white blood cells in the bone marrow called plasmids of amyloid deposition. In ATTR amyloidosis results from TTR amyloid building up in the hearts of the elderly, whether caused by genetic factors or clinical practice. Yet, for example, because TTR amyloid is often undiagnosed in patients with heart failure, it's very likely that the disease is much more prevalent than now recognized. The symptoms of amyloidosis are usually vague and non-specific, such as gastrointestinal or liver disease with no obvious enlarged liver without alcohol consumption or other. The gold standard for detecting amyloid deposits is to employ Congo red staining on a tissue sample. The easiest